Welcome to this webinar in which we're reviewing the standard gene level results that come from doing the analysis of the human transcriptome array in the Transcriptome Analysis Console or TAC software. The human transcriptome array, also called the HTA array, analysis is performed by two separate software packages at Affymetrix. The first, Expression Console, handles producing the gene level signals or the signal estimates, and also functions as a quality control reporting tool so you can evaluate the performance of the arrays. The second software, the TAC software, or Transcriptome Analysis Console, which we'll be talking about today, does two things. It performs the differential expression analysis at either the exon or the gene level, and also does a similar thing for alternative splicing. Today we'll be reviewing results for the gene level analysis. When you do the gene level analysis in TAC, there's several metrics that are calculated or numbers that are calculated for you. For each condition pair, and then for each probe set, we'll calculate the average standard deviation and fold change. In addition, a one-way between subject ANOVA will be performed on all the probe sets, calculating an ANOVA p-value, and this will also be subject to an FDR correction. So we'll have both the FDR and the ANOVA p-values available. In addition, if there are greater than two conditions, 3 or more obviously, then we'll compute the ANOVA and FDR across all conditions for the probe sets in addition to each of the pairwise conditions. The table will indicate the one for the across ANOVA p-values. We'll be using a gray background to differentiate them from the standard condition pair results. The results that are produced will be displayed in a couple of different things. First, there's going to be a summary, which is a breakdown of the differentially expressed genes, just based by number. Second, there'll be a results table that has the by-weight averages, the standard deviations, and fold changes and p-values as referred to before. In addition, it will be merged with gene level annotation information like gene symbol and gene descriptions. Then there will be some graphs that are provided, including a scatter plot, volcano, and a chromosome summary, and some hierarchical clustering capabilities. One thing I want to point out here before we move on is that the default filters for the graphs and the tables are genes will be displayed if they have a greater than twofold change either up or down and a p-value from the ANOVA results of 0.05. Okay, so now we'll do a transition to a live demo of the TAC software. Okay, so here's the results after a standard analysis has been run for the gene level. On the left, we have the summary table, and on the right, we have the default view of the scatter plot. If you have a small monitor and you prefer to have only a single view of a single view at a time, for instance, just the summary table or just a graph, you can turn off the split view by clicking this box right here, which makes it go to a single thing so that your table or the scatter plot is displayed only, and that's all you see. I prefer to have both next to each other, so here we have the, which is especially true once you get to having the data table and the graph next to each other, but we'll get to that in a second. So here's a summary on the left, and you can see we have a breakdown of the total number of genes on the array, a little bit over 70,000 genes, broken down into both the coding and the non-coding. Then you can see that there were about 6,500 genes that were found to be differentially expressed, 4,600 of which were coding and 1,500 non-coding. The other refer to some controls that are on the array. Then we have further breakdown by the number that are upregulated in muscle and the number that are downregulated in muscle. Summary of the algorithm options that were performed, and here is a breakdown of the default filtering criteria and the assignment of the arrays to the various conditions is shown here. If we click on here, we can now see that we have the table displayed on the left, which contains all of the significant values or numeric data associated with it, including some of the annotation information that's been fused on. And on the right, we have the graphical displays. The first thing to know is that both the graphs and the table are dynamically linked to each other. So if we highlight a set of probe sets here in the graph, they are also highlighted in the table. Okay, and vice versa. If we highlight things in the table, they're highlighted in the graph. So once you have a set of things highlighted here, if you want to move to ones that are off the screen, you can use the arrows that are located down here to move down through the list or back up through the list in order to navigate. Okay, and then if you're interested in a particular gene that you have found here but you're not familiar with, you can right click on it, which will enable you to link out to either NCBI, Ensemble, UC Santa Cruz, or to NetFX available from Affymetrix.com. So if we go back to the table here, we can see we have a set of genes on the left and we have a scatter plot on the right, which is basically the tissue versus tissue, the signals in tissue versus tissue. Okay, if you want to clear your selection, you can simply hit the clear selection button here. In addition to the scatter plot, we also have a volcano plot. One thing that I'm going to point out here is you'll note that at the bottom the x's are small, and as you move to the top they get larger. This is because the size of the x in both the scatter and the volcano plot are related to the significance. So the lower the p-value, the larger the x. And so this is a standard volcano plot, so along the x-axis we have the fold change, and along the y-axis we have the significance. An interesting view that is also provided in the transcript analysis console for the gene level results is this chromosome summary. And so you can see here that all of the genes that are selected over here on the left are highlighted in blue over here on the right. And so you can see that there are a number of genes here on the top on chromosome 1 and chromosome 2 that have been selected. If you look very closely, you'll note that none of the genes on the bottom, or none of the green track, have been selected, but that's not surprising, because if you remember when we made the selection back here on the scatter plot, we only selected genes that were upregulated in muscle. Okay, so if we clear this selection, you go back to the chromosome summary now that is clear. So in addition to these three views, we also have a hierarchical clustering function. One thing with the hierarchical clustering, though, is that it's limited to the number of genes that it can handle, and you need to have about less than 5,000 genes. Well, this is okay, because now I can give you an example of how we can go about adjusting the filtering and the display on the table on here on the right. So if we go over here, we can look and see that by default the standard sorting based on fold change and the fold change cutoff and the ANOVA p-value have been implemented. But let's say we want to adjust the filter on an FDR also. So then if you go to the column header and you right click, it brings up this pop-up menu. You can select filter, and now you can choose to enter a value which to filter on. So let's do 0.001. Okay, and select OK. You can see that we've not reduced the number of genes very much. So in addition to this, let's also go ahead and adjust the fold change. So we'll right click on the fold change and we'll look for things that are only expressed by large amounts. So let's go less than or greater than 6-fold and less than -6-fold change. Okay, so now you can see we're down to about 1,300 genes, and so we're able to perform the hierarchical clustering. So let's go ahead and hit that, and after a few short seconds the results are displayed here on the right. Okay, and it's a standard heat map of the hierarchical clustering results. Because I am colorblind, I have adjusted it so that it is the red versus blue, but if you right click on this, you can see that it's possible to change that to the more standard red versus green, or a simple gray scale for the hierarchical clustering. So there's two pieces of functionality here that I want to show you within the hierarchical clustering results. The first is that you have the ability to zoom in to a particular region, and that can be done in two ways. The first way is you can use your mouse and click and drag a little box, which will do a selection, and will zoom into that selection. If you want to further zoom in, over here on the right are two sliders. One which allows, if you grab it, allows it to zoom in as you move it down, and zoom out as you move the other direction with it. And then there's a scroll bar so that after you've zoomed in you can navigate, you can scroll up and down to see the various things. The other thing you'll notice that has happened after you've zoomed in is that now we're able to display the transcript cluster ID and a gene symbol if it's available for each of the rows, so that you can start to look at the gene symbols that are present within a particular cluster. If you're interested, you can navigate around here until you get a set of gene symbols or genes that are of interest that are available, and then right click on the graph, and then you can export these data either to a text file or to the clipboard so that you can copy them in another application and paste them in there. In addition, it's possible to print and save the picture for to a PNG, and these options are available also in the chromosome view, the scatter plot, and the volcano plot. So that basically ends the tour through the gene level analysis in the TAC software. Before I leave you, though, I want to take you back and point out a couple of other available resources that are available on Affymetrix.com related to TAC and the Expression Console software and the ability to analyze the HTA arrays. The first is the Net Effects Analysis Center, in which you can get probe set information. The second is the software manuals for both Expression Console and TAC are available, in addition to several white papers and other data sheets. And then there are further training videos that I've made, or that other people have made, related to Expression Console and Transcript Analysis Console. So thank you for your attention.